Chapter 14, Section 5, Coordinated Regulation of Glycolysis and Gluconeogenesis. Now we have to keep in mind, it's not a one or the other type of thing when it comes to either the cell is going to do glycolysis or it's going to go do gluconeogenesis. And it's completely separate and all things, this cell can only do this one, this cell can only do that one. Turns out it's more of a teeter-totter effect. Glucose goes through glycolysis for right now, but things change. And for the next hour, a few minutes, hour, maybe a couple hours, we're going to go through gluconeogenesis because we need to increase, our glucose concentration in the cytoplasm so that we can go through and make some things or release the glucose into the bloodstream because our blood sugar levels are dropping. Okay, so simultaneously you can end up with both of them, like I said, on a teeter-totter thing. Now we're going to do this, we're going to switch over, now we're going to go back, now we're going to do this, now we're going to go back. By having it so it's more this threshold level teeter-totter effect, evolution has made this so there's minimal waste. Yes, there's going to be some waste, it just happens. But if it was an all or nothing, here's what we're going to do now, and the other cells will do this, and you have too many cells working against each other and lots of waste. Excuse me. Now, if you remember from previous sections, talking glycolysis, hexokinase gave us glucose 6-phosphate. Talked about the fact that there were 6 hexokinases. 1, 2, 3, and our friend 4. Now it turns out that this is again not an all or none. You can't just have these things going full tilt if they're there and then have to get rid of them, and then whenever we need to go through glycolysis again, remake these proteins. No. Our cells make them, maintain them, and then just judiciously regulate when they can be active and when they can't be active. Turns out hexokinase 1, 2, and 3 are all inhibited by glucose 6-phosphate. So again, it's a threshold thing. High levels of G6P, glucose 6-phosphate in the cytoplasm, hexokinase 1, 2, and 3 are gonna be slow and sluggish in their activity rate. But as those levels drop and get below a threshold, well, the activity rate of hexokinase 1, 2, and 3 will increase. When G6-3P goes back up again, they slow down. When it drops, they speed up. So you get this, they're always there. It's just how quickly they're working will be variable. Now, hexokinase 4, also known as a glucokinase, this is a liver enzyme that is not inhibited by glucose 6-phosphate. Glucose, or hexokinase 4, the glucokinase, has a different role. It's not so much, hey, let's prepare glucose for glycolysis. This one is more in maintaining homeostasis, glucose levels in the blood. You see here, hexokinase 1 very quickly hits its maximal and maintains, no matter what, how much you increase glucose. Whereas hexokinase 4, no, gets pretty high up there before it hits max. It's Vmax, well beyond what you would not see in normal blood levels. Here, regulation, yes, no, can it be active, is not going to be a, inhibition by a product, its product, or a later enzyme in the pathway's product. Instead, it's going to be sequestration. Hexokinase 4 is going to be stored in the nucleus until glucose enters into a hepatic cell. It's going to interact with the transport enzyme proteins, and it's going to allow for hexokinase 4 to be transported out into the cytoplasm. Here in the nucleus, it's held by a regulatory protein kept in stasis. Glucose enters into the cytoplasm, and you see the release of hexokinase 4. Hexokinase is going to take glucose, turn it into glucose 6-phosphate, fructose 6-phosphate, the next step, by a different enzyme. This enzyme here inhibits this product here, later product. Well, this is an effector. This slows down the activity of and allows hexokinase 4 to be taken back into the nucleus. Phosphofructose kinase 1. Okay, remember this is the enzyme that takes fructose 6-phosphate and turns it into fructose 1,6-bisphosphate. This enzyme is one of the major steps in glycolysis. Its reciprocal in gluconeogenesis is pushed by fructose 1,6-bisphosphatase. Remember, this takes off the phosphate group on carbon 1, releasing that inorganic phosphate. So these are working in opposite directions from each other. Turns out high levels of ATP inhibits PFK1, which is involved in glycolysis, which makes sense. If you already have high levels of ATP, no reason to go through glycolysis to make more. You're not using what you have. And there's only a finite amount of a diphosphate to be turned into ATP, a triphosphate. So if you already have above a certain threshold, almost maximum, why go through all the waste? Why go through all the trouble of allowing glycolysis? If the ATP is you still have some there and it's not high levels, but it's still interfering, turns out a diphosphate, a monophosphate push. They alter, they counter the allosteric inhibition effects of a triphosphate. So you can look here, binding site for a diphosphate, allosteric regulator site for a triphosphate. So they can work against each other, as you see here. Going from fructose 6-phosphate to fructose 1,6-bisphosphate, which is what PFK1 does. ATP inhibits, AMP ADP push through. Turns out we also have some others. If you have high levels of citrate, it means that you've made a lot of pyruvate that's going into the citric acid cycle and ultimately will help generate a lot of ATP. Why keep making more pyruvate to feed into the citric acid cycle when you have enough? So citrate is also going to stop this step. Signal molecule we're going to be talking about in a few slides. Fructose 2,6-bisphosphate, not 1,6 like we have here, but 2,6. This is going to be used in glycolysis. This is a signal molecule. High levels of this push this change. It wants the cell to go through, almost wants the cell to go through. Citrate, as I said, counters this. If you've got enough going into the citric acid cycle, you do not need more. So high levels of citrate means that the citric acid cycle has what it can handle. Why keep making more pyruvate? Why keep doing it? You've got enough there. You've got a backup of what's there. So let those levels of citrate drop as the cycle goes through more. Then there's nothing else that's allosterically inhibiting PFK1. Glycolysis can resume until you get high levels of citrate again, and it just seems to be this, like I said, it's a threshold, a teeter-totter thing. Got too much, stop. Don't have a lot, go. So when we look at PFK1 pushes through glycolysis, fructose 6-phosphate to fructose 1,6-bisphosphate. The counter to this, remember taking, in gluconeogenesis, taking fructose 1,6-bisphosphate and going backwards to fructose 6-phosphate, releasing an inorganic phosphate. That was done by fructose bisphosphatase 1. Low levels of AMP. If you have high levels of AMP, which means you're using up a lot of ATP and stuff, you're losing a lot, cyclic AMP levels are high, whatnot. Well, you need to replenish. You need to go from AMP back to ATP. Means your ATP levels have dropped. AMP levels are too high for that cell to maintain. Stop this. Do not do this. You do not want to take what little fructose 1,6-bisphosphate you have and turn it back into fructose 6. No. If your levels are low of or are high of AMP, so you have high of this, which means you have to have low ATP, then you want glycolysis to resume. You want fructose 1,6-bisphosphate. You want fructose 1,6-bisphosphate to become G3P, to become pyruvate, and enter into the citric acid cycle and oxidative phosphorylation so you can get this back up. On the flip side, though, if it's the opposite, you have high levels of ATP, you have low levels of AMP, boom, go back. Okay, you're good. Cell's good for right this moment. Maybe, signals come through, glucagon. Hey, need to get some more blood sugar out into the blood. Okay, let's take, stop glycolysis and reverse everything. Get it out. Cell's happy. It's got high levels of ATP right now, low levels of ADP, low levels of AMP. We're good. Send it out. As I just mentioned, we have two signal molecules, two hormones that are in our blood that are counter to each other. They're antagonistic in their effects. First one's glucagon. Glucagon tells the liver produce and release glucose. So basically, glucagon promotes gluconeogenesis. Get some sugar out. Break down the glycogen if you have to. Take whatever you've got there in the cell, convert it back to pyruvate, take the pyruvate, convert it back to glucose, and get it into the blood. On the flip side, you have insulin. Insulin tells the liver, yeah, we got enough, get the sugar out of the bloodstream, get it into the cell, start using it. Make glycogen if you have to, or break it down and use it in glycolysis. One of the side effects in this glucose insulin, one of the things we have in this antagonistic battle between glucose and insulin is a signal molecule known as fructose 2,6-bisphosphate. Notice, 2, not 1. Fructose 1,6-bisphosphate is in glycolysis. So this is C2 right there, C1, which is the fructose 1,6-bisphosphate. Remember, should be up in the up orientation with the phosphate group, up in the up orientation with the phosphate group coming off of it there. Now here, it's below the plane of the sugar ring. The phosphate group, which is now directly attached to carbon 2, is above the ring. Its job, fructose 2,6-bisphosphate's job, is to mediate glycolysis, gluconeogenesis, glycolysis, gluconeogenesis. It binds to PFK1. Okay, remember PFK1 is the one that's in glycolysis, taking fructose 6-phosphate and turning it into fructose 1,6-bisphosphate. This signal molecule increases the affinity of that enzyme, PFK1, to its substrate. It wants it to work better. It wants it, or it wants to help it. It's a little helper. It's all good. On the flip side, it reduces the affinity of FBPase1. Remember, FBPase1 is the enzyme that takes fructose 1,6-bisphosphate and cuts off that phosphate group attached to carbon 1, releasing it as a free inorganic phosphate. That's remember that non-reversible reaction? Well, here, fructose, good, bad. So, makes this one the good one, makes this one the bad one. So if you have high levels of this signal molecule in your cell, PFK1 good, FBPase 1 bad. And they've studied this. They've shown PFK1 signal activity. Remember I told you that's the one it wants to push through. If fructose 2,6-bisphosphate, which they're showing here as F26BP, if it's present, the activity of this enzyme is super high. If it's absent, it's super low. So remember, it's promoting it, it's helping it. On the flip side, FBPase1 activity. If F26BP is not there, its activity is high. If it's there, it's nice and low. Because when you look at it, again, here's our signal molecule. Remember the one that's going to bind and allosterically help or allosterically hinder. It helps PFK1. It hinders FBPase1. Now, where's this coming from? Well, PFK2, phosphofructose kinase 2, takes fructose 6-phosphate and turns it into our fructose 2-bisphosphate. F26BP is what they're using the acronym for. It just does not roll off the tongue. So PFK2 says yes, make more, where you also have a phosphatase that says get rid of it, break it down. So you look here, here's this, PFK2 takes this, makes the signal molecule, whereas FBPase2 takes the signal molecule, turns it back into fructose 2-phosphate. So that way it can't help, it also can't hinder. So depending upon what levels, how much is there? High levels of this, of the F26BP, means we're looking at glycolysis. Low levels means we're now looking at gluconeogenesis. So here's what's controlling our teeter-totter, or at least one of the things controlling our teeter-totter. Glucagon. Okay. Glucagon. Remember, this is the one that's going to tell the cells release glucose into the bloodstream. Blood sugar levels are getting too low. Glucagon, via the making of cyclic AMP, okay, and then there's a cyclic AMP-dependent protein kinase, which gets activated by high levels of cyclic AMP, calls for the breakdown of fructose 2,6-bisphosphate. Okay, remember I just talked about it. Low levels of F26BP means no to the glycolysis, but yes to the gluconeogenesis. On the flip side, insulin. Goes through, gets rid of this phosphorylation event due to the cyclic AMP-dependent protein kinase, which means that the levels of F26BP, fructose 2,6-bisphosphate, will increase. Thus, we see the push towards glycolysis and the inhibition of gluconeogenesis. Another regulatory molecule we see here is what is known as Xylose 5-phosphate. Okay, our pentose phosphate pathway comes into play here. Our phosphoprotein phosphatase 2A is the other side of the coin. One is going to slow things down, the other is going to increase our little signal modulator. So we have a regulatory mechanism on top of for our regulatory mechanism. So in the glucagon, it's coming through, saying ATP to ADP because of the protein kinase A. Pyruvate kinase L becomes inactivated because it's been phosphorylated, but it gets the phosphatase removing it. You now have an active form, the active form which will then go through and phosphorylate PEP, help with this to remove PEP and then turn it into pyruvate. Pyruvate, which through some different things can become alanine. Alanine can be transaminated, remove the amino group, turn it back into pyruvate, and all sorts of fun stuff can happen. If you have high levels of acetyl-CoA, not just the mere presence of here's a molecule, but remember a lot of what we're talking about here in metabolism, it's not just a yes or no, is it there, yes, no, on, off, but a threshold event. Have high levels of acetyl-CoA, then this gets inhibited. No reason to do it. High levels of long-chain fatty acids. No reason to do it because those long-chain fatty acids are going to be cut, turned into acetyl-CoA and then fed into the citric acid cycle. So why waste going through all this stuff in glycolysis? So if you have high levels of fatty acids present, it means you don't need the energy from the sugar molecules. Those sugars can either be stored as glycogen or used somewhere else in the cell for something different. So if you have lots of fatty acids that are being turned into acetyl-CoA, acetyl-CoA goes and stimulates pyruvate carboxylase. Remember, that's our workaround, going back from pyruvate to phosphoenolpyruvate via the oxaloacetate, entering into gluconeogenesis. Have high levels of this, you don't need phosphoenolpyruvate being turned into pyruvate. Because the pyruvate is going to be turned into this also, and you're going to take what's already at a high level and take it up even further. So you have insulin dumping into the blood, whether it's from a needle, whether it's from your own cells. You're going to have increased expression of hexokinases, phosphofructokinase, PFK1, which we just talked about, certain dehydrogenases, and you're looking for increase in glycolysis because you got a bunch of sugar in the bloodstream and you needed to get it into the cells and utilized. So our friend, I'm not even going to try to pronounce this acronym, carbohydrate response element binding protein. It's carbohydrate, CHREPD. This is a transcriptional factor in liver, adipose cells, adipose tissue, because remember, this is where you're going to be storing lots of your excess energy, and in the kidneys. So glucose uptake via GLUT2, glucose hexokinase, because it's released from being sequestered in the nucleus. Glucose, glucose 6-phosphate, step, step, step. You have here Xylose 5-phosphate, which will activate our response element binding protein, which dephosphorylates this, allows it to be active, then translocates to the nucleus, where it's going to be dephosphorylated once again via PP2A, because high levels of Xylose 5-phosphate are going to activate this phosphatase, pulls off one, activates again, pulls off the other, and you now have what wasn't it double phosphorylated, and in the inactive form, you now have in the active form, and it will bind, and then you'll get the expression of sets of genes that are then responsible for glycolysis, which makes sense if you're taking up glucose. Here's a whole bunch of new enzymes and stuff that are going to help upregulate, help glycolysis go through, are going to aid allosterically help in glycolysis. And then you're going to have some that are going to be there for fatty acid synthesis. You're going to have lots of pyruvates, lots of acetyl-CoAs are going to be made in excess, so pull off that acetyl, start linking together 2 carbons by 2 carbons to 2 carbons, the next thing you know, you got a fatty acid. You can start using those to build other things.